Good morning, everyone. I am Naratippakorn from Department of Periodontology, Faculty of Dentistry, Chulalongkorn University. Today, I would like to present our research, expressions of vascular endothelial growth factor protein in mRNA-transfected human periodontal ligament cell. Periodontitis is a chronic inflammatory disease which results from host immune response to dental plaque. The disease destroys periodontium to supporting structure, including gingiva, periodontal ligament, cementum, and alveolar bone. If left untreated, it could lead to tooth loss, which affects how to eat, speak, appearance, and the quality of life. In this study, we would like to focus on periodontal ligament. Initial therapy for periodontal treatment consists of removal of dental plaque and calculus by scaling and root penning. In the advanced forms of periodontitis, surgical procedure utilized for periodontal regeneration, including guided tissue regeneration and bone grafting. However, this treatment provides unpredictable with mixed clinical outcome and costly. Periodontal regeneration is based on the periodontal tissue engineering strategy, which includes stem cell, scaffold, and growth factor protein in the context of an adequate blood supply. Growth factor protein for clinical use consists of pellet-derived growth factor and bone morphogenic protein too. Our study is interested in vascular endothelial growth factor or EGF. However, half-life of growth factor protein is short, so supraphysiologic dose are required. Such high dose of growth factor may cause undesirable side effects and increase the cost of treatment. Therefore, gene therapy may provide a better bioavailability of growth factor. Gene therapy involves the delivery of nucleic acid, either DNA or RNA, encoding growth factor into patient cell, and the cell becomes the growth factor protein factory. mRNA is a novel technology platform for vaccine and therapeutics. It has proven to be a successful vaccine modality against COVID-19, demonstrating safety and high efficacy in humans. mRNA transfected the cell by endocytosis and translated into protein in cytoplasm. In fact, the major limitations of using mRNA were its highly immunogenicity and instability. This obstacle has been overcome by modifications of mRNA structure, replacing uridine with pseudouridine. As a result, it reduced immunogenics and enhanced protein expression. mRNA also needs a delivery system encapsulated by lipids that can prevent RNA's digestion and enhance mRNA stability. Overall, modified mRNA-based technology has advantage in terms of safety, since mRNA do not enter the nucleus, no risk of mutagenesis, lack of inflammatory response, stability, rapid and high protein expression, and simple method. Our research would like to use mRNA technology for periodontal tissue regeneration. The specific subject of this study are first to evaluate vascular endothelial growth factor protein production from culture supernatant of pseudouridine modified mRNA encoding VEGF transfected periodontal ligament cell or PDL cell. And second, to evaluate angiogenic effect of translated protein using CAM assay. Material and method for in vitro study. First, PDL cells were harvested from extracted tooth due to orthodontic purpose at Faculty of Dentistry, Chulalongkorn University. Then isolate PDL cells were transfected with pseudouridine modified mRNA encoding VEGF obtained from Thailand biotechnology and complex with transfecting agent lipofectamine 2000 or L2000 as a control. After 24-hour culture, supernatant were collected and VEGF protein was measured by ELISA. In addition, cell viability was assessed using ALAMA Blue. Here is the result of VEGF protein production. We found significant higher VEGF protein production in mRNA on the black bar than L2000 control group, the white bar. The mean VEGF in mRNA group was around 25,000 picogram per milliliter, whereas like only 500 picogram per milliliter in L2000 control group. We also found good cell viability, which is greater than 85% in both mRNA and the control group. For discussion, our in vitro data using modified mRNA encoding VEGF in L2000 supports previous data in our laboratory that use other growth factor mRNA such as PDGF and BMP2 with similar culture condition or reported high protein expression after 24 hours. PDL cell transfected with corresponding growth factor modified mRNA that complex with transfecting agent L2000. Next, we investigate in vivo the biological functions of translated protein in PDL culture supernatant collect from the in vitro study. We use CAM assay to measure angiogenesis, or in other words, blood vessel formation. CAM assay has been a well-established model for studying angiogenesis due to its simplicity, rapid development, and cost-effective. In this study, 15 white leghorn chicken eggs were obtained from Kasetsart University. The day we received those eggs is day 0 in CAM assay. All the eggs were incubated at 37.5 Celsius with 60% humidity. After 3-day incubation, we need to separate CAM from the eggshell by aspirating 3 milliliters of albumin and make a small window and cover with a sticky tape and then re-incubate till day 8. On day 8, filter paper will with 5 millimeter diameter load with the substance of interest, that is PDL culture supernatant mRNA group or L2000, and we use DPBS as a negative control and further incubate for another 3 days. So on day 11, all the eggs were terminated. To evaluate at the angiogenic response, we count the increased number of blood vessels that convergent to the filter paper under stereo microscope between day 8 and day 11 and compare the increased number of blood vessels between groups. Here is the result, the CAM result under stereo microscope. They are representative from each group, DPBS, L2000, and mRNA group on day 8 and day 11. As you can see, more microvascular network as a spoke wheel pattern, or the black arrow that in the picture we observe in all groups on day 11, but obviously more abundant blood vessel in mRNA group than L2000 group and DPBS group. For counting the number of blood vessels, the corresponding image, we use image J as down below. You can see in the graph plotting the average number of increased blood vessel and individual group. mRNA group shows significant increased number of blood vessel to about 50% of 50 blood vessel, while there are only around 30 in L2000 and DPBS group. For discussion, our result is corresponding to Ramne and colleague in 2019, which they used VEGF mRNA-transfected in osteoblast light cell on polymer scaffold. They show that VEGF mRNA has the ability to induce significant number of blood vessels in CAM assay. In conclusion, we demonstrate that modified mRNA encoding VEGF can promote VEGF productions after transfected in PDL cells with negligible effect on cell viability. This translated protein has angiogenic property to promote the formation of blood vessel in vivo. For the next step in the development of this mRNA therapeutic platform, we would like to further evaluate the ability of VEGF mRNA to promote soft and bone tissue regeneration in a larger animal model. Thus, this mRNA could be beneficial in the treatment of periodontal regeneration. I would like to thank for the support from Center of Excellence in Periodontal Disease and Dental Implant, Thailand Science Research and Innovation Fund, Chulalongkorn University, and the Program Management Unit for Human Resource Institutional Development Research and Innovation. Thank you.
Thank you so much, Dr. Mana. It's quite interesting study. Is there any question from the audience or any comment?
Hello.
Please.
I have a question. Thank you for the good presentation, Dr. Mana. I'm Chirapat from Chulalongkorn University. Your expected result is angiogenesis, right?
Yes.
My question is, instead of the VEGF, are there any growth factor that encoding to the mRNA that provide the angiogenesis outcome?
So besides
another growth factor.
So besides VEGF, like pellet-derived growth factor or PDGF would be another growth factor that plays significant role in blood vessel formation. So I think we can like do more experiment on that growth factor too.
And just one more question. Are there any limitation of this study due to study with the chicken egg? Are there any limitation in animal study?
Yeah, so whenever the embryo is getting bigger, or the embryo move to the area that I put the scalpel, when I took picture, sometimes it's really hard to count when the embryo is underneath. So sometimes I have to exclude that subject out. Yes.
Thank you for your answer.
Thank you for the question. Is there anyone else have a comment or question? So I have one question, Dr. Mana, about the CAM assay. Do you have? Is there any other test that can use instead of CAM assay to evaluate the blood vessel, the angiogenesis?
Yeah, so actually actually there are many angiogenesis model that we can use, like the cornea or on the like a bigger and bigger I mean the orchid, the egg orchid that we can use as well. But the reason that I choose this one is because the chicken is faster development because it's small, and we take less time than we use larger animal. So I choose this one. Yeah. And
Thank you very
for the
much.
cornea, yeah.
Okay, you can go further.
For the cornea, I saw in the paper, it's quite scary for me because actually it's really easy to see the result, but I think it's maybe more complicated, and it's quite scary.
I see. Okay. Thank you very much.